Learning objectives. In this module, you will be able to summarize the principles of nucleic acid hybridization. Principles of nucleic acid hybridization. In a double-stranded DNA molecule, the hydrogen bonds between paired bases act as a glue that holds the two complementary DNA strands together. Two hydrogen bonds form between A and T in each AT base pair, and three hydrogen bonds hold G and C together in each GC base pair. A region of DNA that is GC rich is therefore more stable than a region that is AT rich. Each hydrogen bond, although individually weak, but when base paired, extending over many base pairs, results in a cumulative strength of the hydrogen bonds that is quite strong. The common product used in fabric, Velcro, uses the same principle. A single Velcro hook and loop attachment itself is very weak, but thousands of them together make for a very strong fastening system. Double-stranded DNA can be subjected to different treatments that result in the breakage of the hydrogen bonds so that the two strands are separated. This process is known as denaturation. For example, if we heat DNA to a high enough temperature or expose it to a high concentration of chemicals such as formamide or urea, the hydrogen bonds break and the two complementary DNA strands will separate. Subsequently, gradual cooling or removal of the agent, denaturation agent, allows the separated DNA strands to come together again, reforming the base pairs in the correct order to restore the original double-stranded DNA. The association of two complementary nucleic acid strands to form a double-stranded nucleic acid is known as nucleic acid hybridization. Under experimental conditions, two single nucleic acid strands with a high degree of base pair complementarity can be allowed to hybridize to form an artificial duplex. This artificial duplex is known as a heteroduplex. The formation of artificial duplexes, almost always heteroduplexes, is the essence of nucleic acid hybridization assays that are widely used in molecular genetics. An annealing of homologous DNA molecules. Panel A depicts denaturation of homologous DNA molecules. In A, denaturation means breaking of the hydrogen bonds in a double-stranded duplex nucleic acid and can be achieved by heating or exposure to highly polar chemicals such as urea and formamide. Under certain conditions, the separated strands can reassociate or hybridize or reanneal to reconstitute the original double-stranded DNA. In B is shown artificial duplex formation between homologous sequences. In strands that have very similar nucleotide sequences or two different DNA sources that have been denatured and then mixed, for a proportion of the denatured DNA molecules, the original double-stranded DNAs reform, forming a homoduplex. But in other cases, artificial duplexes between the partly complementary sequences form, and these are referred to as heteroduplexes. This forms the basis of many hybridization-based assays used in human genetics. Hybridization assays. Nucleic acid hybridization assays exploit the specificity of hybridization. Two single polynucleotide, DNA or RNA, or oligonucleotide strands will form a stable double-stranded hybrid. only if there is a significant amount of base pairing between them. The stability of the resulting duplex depends on the extent of base matching. Assay conditions can be chosen to allow perfectly matched duplexes only, or to allow degrees of base pair mismatching. Hybridization assays can be performed in many ways, with multiple applications in both research and diagnostics. There is a common underlying principle in that a known, well-characterized population of nucleic acid molecules or synthetic oligonucleotides is used to interrogate an imperfectly understood population of nucleic acids. The known, well-characterized nucleic acid sequence is referred to as the probe, whereas the imperfectly understood population of nucleic acids, the test sample, is known as the target. A hybridization assay can be used to identify nucleic acid sequences that are distantly related from a given nucleic acid probe. Be useful, for instance, in a situation where we are trying to identify related genes. We might want to start with the DNA clone from a human gene and use that as a probe to identify the corresponding mouse gene. The human and mouse genes might be significantly different in sequence, but if we choose a long enough DNA probe and reduce the stringency of hybridization, stable heteroduplexes can be allowed to form even though there might be a significant base pair mismatch. Conversely, we can choose hybridization conditions to accept only perfect matching. This is known as high stringency. If we choose an oligonucleotide probe, we can use a high hybridization stringency so that only probe test duplexes that form are ones that contain the exact same sequence as the probe. That can happen because of a single mismatch out of, say, 18 base pairs can make the duplex thermodynamically unstable. Oligonucleotides can therefore be used to identify alleles that differ by a single nucleotide, otherwise known as allele-specific oligonucleotides. Classes of hybridization assays. There are many types of hybridization assays, but they all fall into two broad categories. In one case, the probe molecules are labeled, and the test sample molecules are unlabeled. In that case, the probe is often a single type of cloned DNA, and it is usually labeled using a polymerase to synthesize a complementary DNA or RNA strand in the presence of one or more labeled oligonucleotides. The alternative type of assay uses unlabeled probe molecules, and it is the test sample molecules that are labeled. Microarray hybridization allows for large-scale parallel hybridization to immobilized probes. The innovative and powerful hybridization technologies developed in the early 1990s permit numerous hybridization assays to be conducted simultaneously on a sample under the same conditions. A DNA or oligonucleotide microarray consists of many thousands of millions of different unlabeled DNA or oligonucleotide probe populations that have been fixed to glass or other suitable surfaces with a high-density grid format. Within each grid square are millions of identical copies of just one probe. A grid square with its population is called a feature. Because the intensity of each hybridization signal will reflect the number of labeled molecules that have bound to a given feature, microarray hybridization is used to quantitate different sequences in a complex test sample population, such as samples of genomic or total cellular RNA. Popular hybridization assays. As stated previously, there are two broad categories of hybridization-based assays. The first involves a labeled probe and then an unlabeled test sample, as shown in figures 3.6 and 3.7a of Chapter 3 of your textbook. The labeled probe method includes the following hybridization assays: southern blot, in tissue in situ hybridization, and chromosome in situ hybridization. In southern blotting, one is looking for medium-sized changes in the order of magnitude of hundreds of base pairs to several kilobases in genes or DNA of a test sample. In tissue in situ hybridization, one is tracking RNA transcripts in tissues and embryos. Potentially, one can be tracking DNA in a particular tissue or embryo. And chromosome in situ hybridization involves studying large-scale changes using chromosomes that have been fixed on a slide as the test sample. So this can reveal many changes that historically were revealed through Giemsa staining in the field of cytogenetics, but now often uses fluorescently labeled probes to reveal abnormalities or changes in chromosome structure. The second type of hybridization assay, that is using an unlabeled probe with labeled test samples, includes the microarray comparative genome hybridization and microarray-based expression profiling. Microarray comparative genome hybridization is useful for genome-wide searches for large mega-base-sized changes in DNA. And microarray-based expression profiling can be used to simultaneously track expression of many genes as occur in a particular disease syndrome like cancer, cardiovascular disease, diabetes, and so forth. Labeling of nucleic acids. Hybridization assays involve the labeling of either the probe or the test sample population. Usually this involves making labeled DNA copies of a starting DNA or RNA with a suitable DNA polymerase in the presence of deoxynucleotides. Different labeling systems can be used, as shown in the table on this slide. Fluorescent dyes, such as the derivatives of fluorescein, are popular. They can be detected readily because they emit fluorescent light of a defined wavelength when suitably excited. Some other labels are detected by specific binding to an antibody or to a very strongly interacting protein. In these cases, the detecting protein is usually conjugated to a fluorescent group, a fluorophore or fluorochrome, or to an enzyme such as alkaline phosphatase or peroxidase that can permit detection via colorimetric assays or chemical luminescent assays. This slide depicts heteroduplex formation in a hybridization assay. A defined probe population of known nucleic acid or oligonucleotide sequence and a test nucleic acid sample population are both made single-stranded and then mixed together and allowed to re-anneal. Many of the fragments that had been previously base paired in the two populations will re-anneal to reconstitute the original homoduplexes. However, in addition, new artificial duplexes will be formed, partly complementary probe and test sample sequences, which are shown in the bottom center. The hybridization conditions can be adjusted to favor formation of the novel heteroduplexes. In this way, probes can selectively bind to and identify closely related nucleic acids within a complex nucleic acid population. In this example, the probe has been labeled in some way, as shown by red asterisks, but in some hybridization assays, it is the test sample nucleic acid that is labeled. This slide demonstrates the concept of hybridization stringency. High or low stringency conditions can be used to detect nucleic acid sequences that are distant related or show perfect base pair matching with a given probe. In any hybridization assay, we can control the degree of base matching between complementary strands in the probe and the test sample. If, for example, we increase the salt concentration and or reduce the temperature, we lower the hybridization stringency. In A, it is shown that in some circumstances, a long probe strand can form a thermodynamically stable duplex with a comparable but distantly related strand within the test DNA or RNA, even though there might be significant base mismatching. Hence, length of probe relates to stringency. In B, alternatively, we can use high temperature and low salt concentrations to achieve high hybridization stringency that might allow only perfect base matching. That is most easily achieved with a short oligonucleotide probe and allows assays to discriminate between alleles that differ at just a single nucleotide position. So low stringency corresponds to a long probe, whereas high stringency corresponds to low salt in the assay conditions and high temperature and a short probe sequence. This slide shows the two classes of hybridization assays. In one situation, we have the immobilized test sample where the probe itself is labeled. On the right-hand side, we show the labeled test sample, which binds to the unlabeled probes that are immobilized. So there's some fundamental differences. On the left, we have the unlabeled test sample that is affixed to the solid support, and we probe that unlabeled test sample with the labeled probe. The probe test sample duplexes form, and then we wash off any non-specifically bound probe, and then ultimately through a series of washings, we finally result with a particular test result. On the right-hand side of the diagram is shown the newer approach, the approach that is used with microarray hybridization assays, where we use a labeled test sample. So the actual labeled test sample is used to interrogate unlabeled probes which are affixed to the solid support. And then the labeled test samples are hybridized to the solid support, and subsequently through a series of washings you finally obtain your final result. Principles of microarray hybridization. A microarray is a solid surface on which molecules can be fixed at specific coordinates on a high-density grid format. Oligonucleotide or DNA microarrays have thousands to millions of different synthetic, single-stranded oligonucleotide or DNA probes that are fixed at specific predetermined positions in the grid. As shown by the expanded item enclosed within dashed lines, each grid square will have many thousands of identical copies of a single type of oligonucleotide or DNA probe, referred to as a feature. An aqueous test sample containing a heterogeneous collection of labeled DNA fragments or RNA transcripts is denatured and allowed to hybridize with the probes on the array. Some probes, for example, the A1 feature, may find numerous complementary sequences in the test population, resulting in a strong hybridization signal. For other probes, for example in the B1 feature, there may be few complementary sequences in the test sample, resulting in a weak hybridization signal. After washing and drying of the grid, the hybridization signals for the numerous different probes are detected by laser scanning, giving huge amounts of data from a single experiment. For ease of illustration, we show test sample nucleic acids with end labels, but sometimes they contain labels on internal nucleotides as well. And this concludes Module 2.